Cool. All right. Well, why don't we start since now it's 6 minutes after
Oh,
people
yeah.
need to be here.
Okay.
All right. So I will welcome everybody to our second to the last event from a busy whale festival month of March. Thank you for joining us on a Friday morning. We're happy to be here with one of our good friends, Mo Flannery. The California Caliber Science has been such a great partner of ours from the day we started. I mean, they realized that this would be a great opportunity to expand the range in which they respond to animals. And Mo in particular and Sue have been so great at helping us develop a response program that Sarah leads now that is really great, and also an education program for the community that we are really proud of. So this partnership has been one of the best ones we've had since we started as an organization, and a lot of that has to do with Mo. She's just an incredible partner. So thanks, Mo, for that. So I'm happy to be here. If you guys have never seen some of these things, it's an amazing treat to get to look at what Cal Academy has in one of the best collections in the world. And Mo is the manager of both the ornithology and mammalogy collections, which are quite impressive. So this is an incredible opportunity to get to see behind the scenes Cal Academy. And it's exciting to note that you guys have opened now to the public. Maybe you can tell Mo, like, is there any kind of special considerations for coming? Do you have to, I imagine you have to make an appointment and such. So maybe you can tell us about that, but it's exciting that you're opening and have some COVID safe interactions with the public. So that's an exciting thing. But the way that we'll do this, folks, if you want to put questions into the chat, we'll ask Mo when it's a good time. And she's going to walk us through some of the collection, and then we're going to take a little bit of a break while she goes to another room, and Sarah will give you a little peek to some of the stuff we're doing in the back room back there. So Sarah, as you know, our stranding coordinator here, will entertain you while we're transitioning with Mo. So I'll hand it over to you, Mo. Thanks so much for joining us this morning.
Great. I'm glad to be here. So just one technical thing that I'm going to do on my screen and you might want to do on your screen is right now I'm seeing everybody. It's great to see everybody. Hello. But you may not want to see everybody. You might want to see just me or the camera that I'm going to show you. So in the upper right-hand corner of your individual screen, your Zoom screen, if you don't know, you can click view and hit speaker view if it's on gallery view. And then it will put me, or in this case, it has Sheila right in the middle of
I just
the screen. I guess it doesn't put myself there for some reason. And then when I switch cameras, it should put that camera. Let's see if that actually happens.
You got to sideways look at barnacles, it looks like.
But is it in the center of your screen now, Sheila?
Yeah.
Okay, because I'm still seeing you.
I'm glad that you get my face. I'll make sure I'm very expressive.
I'm going to try and pin myself.
There you go. Right that's the other thing.
There, because I need to be able to see what I'm showing you, so I don't do things like that. Turn them upside down.
So that's the other thing for folks. If you don't know, in the corner of your own view, or Mo's view, I mean, you can go to that ... and hit pin, and then Mo will always be up there regardless of who's talking. In speaker view, if I start talking, I'm going to show up. But if you want to always see Mo, just pin her view. And also, as a matter of order, let's put ourselves on mute when you're listening, and we won't hear everybody's dogs barking. That's what I say for myself, because that is.
Or as I've done in meetings, yelling at somebody else from in the other room, which is totally embarrassing. I never do. I'm always muted, and then one time I yelled, I wasn't. So we are open. The Academy opened again, thankfully, on March 15th. I have not been on the main floor because as staff, we also have to make a reservation to go spend time on the main floor, just like the public does to visit us. We are currently open at 25% capacity, which is not a lot of people. And actually, San Francisco just moved to the orange tier which would allow us to open at 50% capacity. But for the safety of our guests and for the safety of our staff, we're keeping it at 25% capacity for a little bit longer, you know, till more people are vaccinated and things feel a little bit safer. Socially distance, wear masks all the time, everything we're also used to. No, the food service is only outdoors, and you can't bring any of your own food into the museum to eat in the museum. So hopefully it's going well. And some of our public programming, they've had to reorganize so that, like, you know, the penguin feeding, which is always very popular. We don't want to get a whole group of people there watching and congregating, because then people tend to not socially distance. So we've changed some of the public programming
about how they changed the public programming to deal with COVID right now. They only are going to allow, even though they're allowed.
So now I'm muted too. Sheila, you're muted now too.
I'm muted for the dogs, but I didn't mean to mute you. Sorry.
Oh, that's okay.
Yeah, so everybody, please mute yourself unless we have already muted you.
Yeah. So let's get on with the tour, because that's the really interesting part. So I'm the collection manager for the Birds and Mammals collection here at the Academy. I've been here for 19 years. I also lead our stranding network program, all of our marine mammal stranding, which Sarah and Sheila are a huge part of, covering the northern section of our response range. We are authorized by the National Marine Fisheries Service to respond to dead marine mammals, basically from Año Nuevo down in San Mateo County to Westport in Mendocino County. And Noyo Center covers the Westport down to some parts of northern Sonoma County as well for us. So I like to think of the Noyo Center as a marine mammal satellite group. But you guys do a great job, and someday you'll be on your own as your own stranding network up there. But for now, we all work together, and specimens, some of them come here, and as you know, some of them stay up at the Noyo Center. But most of them are catalog specimens in the scientific research collection here at the museum, which is then available to researchers all over the world. So they can search our catalog online and figure out what kind of specimens we have in the collection. We have 100,000 birds, 32,000 mammals, and 11,000 eggs and nests. We're not going to look at the birds or anything today. That's a fun experience, but that's a whole other tour. We're going to concentrate on marine mammals. And of our marine mammal collection, we have over 6,500 cataloged marine mammal specimens. I would guess we probably have 5,000 that are not cataloged. We got a little bit behind. COVID put us even more behind, and we're short-staffed. So the catalog happens as it happens. But of those 6,500 marine mammal specimens, they represent 49 different species of marine mammals from around the world. The world's largest collection of California sea lion specimens, which is primarily because of Ray Bandar, who is a volunteer who worked with us. He started collecting in 1953. He died in 2017. He collected over 7,000 skulls of everything, anything you can imagine he had in his collection. Something like 300 monkey skulls from the zoo, over 300 harbor seal skulls, over 3,000 California sea lion skulls in his collection. And when he died, that collection all came into the museum here. So we're still working on cataloging his California sea lions. So that'll get done someday. But for now, we have over 6,500 marine mammal specimens and the world's largest California sea lion collection. We also have the world's largest southern sea otter collection. We are the repository for any sea otter that washes up along the coast and gets a necropsy, which is the term for an autopsy, but for an animal. Humans get autopsies, animals get necropsies. So they get a necropsy in Santa Cruz and then they come here. And then we also have North America's largest collection of Guadalupe Fur Seals. We only have about 300, but in 2015, we only had 27. And the rest of them have all been collected since 2015 because Guadalupe Fur Seals are undergoing an unusual die-off event where lots of them are washing up along the entire West Coast, all the way up into Oregon and Washington too. So that's also for another talk. We could talk about unusual mortality events at some other time. That would be great. But I'm going to show you some of our whale specimens just to start because I pulled them out and they're hard to get to and if I have to walk down the aisle. So I'm going to change my video to this camera that I have. Hopefully I won't make you ill while I move things around. Let's start with, so gray whales are one of the species that's also undergoing an unusual mortality event. And we have responded to several dead gray whales in the past couple of years. So this is all gray whale material. And I wanted to show you baleen before we go very far, because we talk about baleen and a lot of the time it's difficult to kind of picture how baleen works. So this piece of baleen is preserved. It's been in our collection for a while. Let me put it back down. This part here, oops, sorry, Jiggles. This part here is the gum line. So this is what attaches to the top of the whale's mouth. And then you can see the individual baleen plates, how they lie close together. So this is the cheek side of the baleen rack. So you have plates that are put together to make a rack. And this is a partial rack. They're usually like 4 or 5 feet long. So the outside next to the cheeks, and then the inside of the whale's mouth, the baleen is frayed to make a sieve mechanism. So when a gray whale sucks up a bunch of mud, they use their tongue to push the water out of the mud and out of the sample that's in their mouth. And all of the food gets caught in the baleen and the water goes out, and then that's how they swallow the food. And baleen is basically keratin, just like our hair. And let's look at a side view of this piece of baleen, because I think you can see really easily. You can see how it's compressed together for the most part, but as the baleen grows, the tips of it get frayed, which is what makes this frayed edge. So you can see all the little tubules of keratin that make up this compressed plate. Now, if we wanted to study where this gray whale was feeding, maybe if he or she was pregnant, if it was a she, a plate like this would give us 3 years of information about that gray whale. You would take a tiny little sample from different sections of the baleen, look at the stable isotopes, which is a chemical that's stored in the keratin, and it would tell you where in the ocean that animal was feeding, which would then tell us what type of food it was eating. And you can look at hormone levels. And so that would tell us over the last 3 years, if it was a female, if she was pregnant, if she underwent some sort of stress of some kind. So gray whales, you only get about 3 years worth of data from a piece of baleen. Blue whales, you can get something like 16 years worth of data. So we can't actually age a whale by its baleen because it gets frayed as it grows. So it basically deteriorates. This is a pelvic bone from an adult male gray whale that washed up in 2019. And this is his ear bone. So when we're out on the beach and when Sarah's out on the beach responding to a whale as a museum collection, we want to have a piece of the animal in the collection as a permanent record of that animal at that place at that time. So with a big whale, it's, you can get baleen. That's great if it's there. Sometimes it has already fallen out. Pelvic bones are a small bone. They're a unique bone to each individual. Some of them are shaped very crazy looking. We've gotten some that have had residual femur bones attached to them. But these are what used to be the whale. Well, it is still the whale's pelvis, but it used to have legs attached to it when whales had legs when they were on land. But it is still a useful bone. In males, the penis actually attaches to the pelvic bone. So for a long time, people just thought, oh, it's just a bone that's hanging out in the whale's body and not serving any purpose. However, a researcher in LA just recently looked at a lot of pelvic bones and determined that they could be part of sexual selection because the penis is still attached to them. That paper is not published yet, and I think he's still doing the analysis, but it's interesting to know that it actually still has attachments and could still be useful. And then the ear bones are also something that can be useful to researchers if we allowed them to take a cross section of the ear bone. You could age the animal based on the layers of bone in the ear bone. They're also useful for individual variation. And then the inner ear bones, several people have studied inner ear bones, which often fall out, but are also useful for researchers. Whales also have something called an ear plug. I've never found one on any whale that I've worked on. I really want to find one. They're really hard to find, but basically it's a tube of wax that builds up inside the ear canal of the whale. And if you can retrieve it whole, you can count the layers of wax just like you count the layers in a tree. And you can also do chemical analysis similar to what I talked about with baleen, looking at hormones and stable isotopes and toxins in earplugs. So we're always trying to find the earplug. I think that by the time we get the animals, they're pretty decomposed. And since it's wax, we think that it melted. That might just be an excuse that I'm making because I have never been able to actually find one and extract it whole. And there are folks that can find them, and especially like during the whaling, when people are harvesting minke whales over in Finland and other places, they find the ear wax, the earplug all the time because it's a fresh animal, I think. So then we also collect parasites. This is barnacles on a piece of gray whale skin. So the skin can also offer DNA analysis. Researchers have also studied the barnacles, taken little small sections to learn where the whale traveled with that barnacle, because the barnacle also holds the stable isotopes in its calcified shell. And so you can, if you examine the barnacle, you can see what part of the ocean the whale was in while that barnacle was growing that layer. So very, very cool. And then on the bottom, I think you can see them. Those are whale lice, which are copepods called cyamids or cyamids. I always forget the right way of pronouncing it, but they are a marine invertebrate that attaches to the outside of whales. It is believed that different species of whales have their own species of cyamids as well. And then the last two items I have to show you are, this is a sperm whale tooth, a complete sperm whale tooth. That's the whole tooth intact. But one of the pieces of research that people can do is they can cut the tooth in half. And so next to that tooth is another tooth from the same animal that was cut in half. And the researcher first counted the layers. So the layers of cementum and dentin are laid down for each growth cycle of the sperm whale. So that tells you how old the animal was. And then you can see there's little drill holes in there, drill lines. we allowed her to take samples from each of those layers and look at the stable isotopes in those layers. And then that's what tells you where the animal was feeding during that period of growth. And I cannot remember because she aged a bunch of animals at the same time. I think that sperm whale was 44 years old. And this is one that washed up in Point Reyes National Seashore with 400 pounds of fishing net in its stomach, which is probably why it died. And we also think it was probably an old gentleman. So 44 is probably pretty old. All right, so I'm gonna move. Now's a good time if anyone has a question to put it in the chat while I'm gaffing my tables.
So Mo, we did the analysis on the killer whale teeth.
Oh yeah.
You remember, I think I say he was closer to 30 than 20. We thought he was 20, and I think with the analysis came out closer to 30.
Yeah, I think that's right. I think that's right on that one.
We also,
I know I have them written down and I put them in the database. It's just one of those things that I just cannot seem to remember.
yeah. We also, I see Jeff joined us, Jeff Jacobson. We've also had
Great.
a researcher doing a radioisotope analysis on the baleen of our blue whale. And so we have some data on where she traveled in the last few years of her life as well. And now apparently she's doing the hormone analysis. So hopefully we'll find out maybe in the next year more about, you know, her history from her hormones. But we do have a little map of where she traveled in the last few years.
Oh, cool. And do you know how many years of data were in there? Was it
16 years from a blue whale plate? But Jeff's just joined us. Maybe he can say, I thought it was 4 years from the plate, but I think we go back 4 years. Maybe she could have gotten more.
Maybe it has to be a really big plate to get the 16 years. Like
Yeah,
if you're sure.
It was
Go
just
ahead,
4
Jeff.
years, 3 to 4 years. And she migrated south from the California coastal current down to Costa Rica Dome area and didn't 1 year.
Huh.
Yeah.
But
I'll send you a link to the paper. It's in +1.
But stayed more along our coastline than we thought. So Leah's asking, what is it about the stable isotopes that tell us where in the ocean the animal is? Can you explain that?
Jeff can probably explain that more because I don't actually, you know, I take care of the specimens and I let the real scientists do the research. Do you want to explain it, Jeff?
I'll repeat what I've been told. Every body, it's a trophic level thing. So that as you move further up the trophic level, the heavier elements, the heavier isotopes of any element tend to move slower out of the tissues. So they accumulate disproportionately. And then that in the body, and then that scales according to trophic level, how far up the food chain you go. So a killer whale eating a pinniped is going to be different than a blue whale eating krill. And then each body of water has its own signature in the krill and the other animals that live in those waters, because that's where those isotopes come from originally, is from those waters. So the study was broad in the California coastal current here, Gulf of California, and then the Costa Rica Dome area. And I just sent Geraldine a whole bunch of samples that we'd collected, along with Karen, one of those, and so that she could ground truth or water truth the stable isotope signature of these waters. And they're now wrapping up, not doing just all the stable isotopes in one big mush, you know, to the signature of all the compounds in the baleen. They're separating it out into compound specific stable isotope, and they're choosing the compounds relative to various metabolic pathways. It's just magic.
That is great.
That's coming out soon.
And that's a really good explanation for it too, in my understanding, and much more well articulated than I could have done. So let me show you another couple of specimens here while we're So we've moved down a row. Ooh, fuzzy wuzzy. We've moved down our main collection storage area of mammals, skulls, and skeletons. I'll just pan around a little bit. If I'm going too fast, let me know. So we organize everything in taxonomic order on the shelves. You can see there's some sun bears there. And on the side where I first came down, we have the beginning of the carnivores. So dogs and wolves. And then we start in on some of the marine mammals. So everyone is organized by who they're more closely related to. So you end up first here with our walruses. That one looks like it's giving us a big hello. I didn't realize it looked like that in the video. And then here's a more adult walrus with those long tusks. And then here's one of our baby walruses. And here on the shelves, these are called baculum. These are walrus penis bones. So this is actually a piece of data that we add to our collection. And we collect the penis bone from every male animal that when we can find it, which hopefully we do most of the time. And that's because that tells future researchers that we identified that specimen to the correct species or to the correct sex. Lots of species like harbor seal. Let me get you a harbor seal skull, which you have probably all seen, but I will show you anyway. We'll just do this one quickly here. So this is a male harbor seal, and the female harbor seal is exactly the same size. There's very little, maybe a tiny little bit of sexual dimorphism in the harbor seals. And so because they're similar size, if we have data that proves that we identified the gender correctly, we want to keep that for future researchers. So they know for sure, oh yes, this is actually a male because it has the baculum in the box with it. Now, some young males look a lot like adult females in some species, like stellar sea lions and California sea lions. You could get them confused if you don't know their teeth very well. And so the baculum is just proof. And in fact, we've looked back at some specimens from 100 years ago that don't have the penis bone in there. And we've actually questioned the gender ID because of the size of the teeth. Like, was this really a male or was this a female? And they got it wrong. Because if you don't find it, that doesn't necessarily mean it's a female. So there's our a nice harbor seal skull. And then while we're down this row, so it, and here, I should have brought this out while I was going back. I'm going to get my steps in today. Here is a harbor seal baculum, right? So very small compared to the walrus baculum, but an important piece of data. Let me grab this guy here. So I would bring out a bigger specimen. You can see the size. It's not as huge as I would like to show you, but I can't get them off the shelf anymore because when the Bandar collection moved in, everything compressed, and the big northern elephant seals are just a little bit too difficult to get off of the shelf without doing some major reorganizing. So this is a subadult male northern elephant seal. You can see where the big proboscis would be on this guy. You can see his really large canine teeth and some small peg-like teeth, which if you ask Sarah, those are a pain in the butt to clean a northern elephant seal and get the teeth in the right place, because they don't fit like a lock and key like most of the other pinnipeds. California sea lions, you put it in a bucket of water, the teeth all fell out. They only go in one way. But in a northern elephant seal, they can go in anywhere. And to be scientifically accurate, you want to get them in the right place. So not a fun job. Well, one of the things that everybody is always interested in, and I wanted to talk about, are the nasal turbinates. So you can see if this guy was laying on the beach, his nose would be sticking out here. And he has all these beautiful layers of bone. They're really quite stunning. Those bone layers are covered with a membrane and with mucus when the animal is alive. We have them in our noses as well, nasal turbinates that are covered with mucus and cilia. And as we breathe in, it cleans our air. And as we breathe out, it helps to absorb some of the water that we would lose through our breath. And that is incredibly important for northern elephant seals. When they are on the beaches molting in the fall, they're just laying around for months and months and months, shedding every piece of their skin. It's the most disgusting thing I've ever seen. And when I've gone on the beach and seen these chunks of elephant seal skin, it's really gross. I don't think dead animals are gross, but I think skin coming off of an animal is gross. Go figure. But anyway, as they're laying there on the beach, they're helping to retain some of their fluid, some of their hydration level. And that's especially important because they're not really feeding during that time period of molt. So they need to stay hydrated. So that's one of our northern elephant seals. This one was tagged. So it does have some green flipper tags that came off of the specimen and then stay with the specimen forever so that you can look back and see maybe where that guy came from, if he was from rehab or if he was tagged on, say, Año Nuevo Island or another rookery somewhere. So let me put him away and then grab one more specimen on this row.
Hey, Mo, while you're doing that, Erica asked about the baculum, and it's a B,
Yeah.
Erica, not a V, baculum, and why? What is it used for and why?
So we think that because marine mammals have to reproduce very quickly, well, carnivores in general. So not all species have them, as you might know. Only several species in the animal world do. Primates, non-human primates have them. Rodents have them. Insectivores like shrews have them. Chiroptera bats have them. And then carnivores. So carnivores are the biggest family of animals that have baculum. And we believe it's for quick mating. Cats and dogs and marine mammals have to do it very quickly. And so it helps. They just don't have the hydraulic system that other species do. And then like the ungulates and the whales and cetaceans do not have them. So which makes sense because whales were more closely related to hippos in their history. And so than they are to other animals that live in the ocean. So that's one characteristic of that family. All right. This is one of my favorite specimens. Back to the camp. So I said we had the largest collection of southern sea otters of any museum in the world. We have over 1,500 cataloged, and we probably have about 1,500 backlogged. Is that focused enough? See if I press the gray button, if it will focus it a little bit more.
It's pretty
a
good.
A little better, huh?
Yeah,
so a lot of our specimens are skulls, like you saw with the walruses and the harbor seal and the elephant seal. And that's because it's a lot easier to pick up a skull on the beach, to collect a skull, to remove a skull, than it is to do an entire skeleton. And if we did every entire elephant seal skeleton, we would not have room in our collection for much of anything else. or whale skeleton. And the skulls are really helpful because they have the teeth, right? So you can study a lot from an animal's teeth. You can look at the wear. You can, like that sperm whale tooth that I showed you was cut in half. You could do the same thing with canine teeth and premolars in this species. They use premolars. In California sea lions, they use canine teeth. But you can also age the animal with their teeth. If you wanted, you could study brain volume and brain size. There's all sorts of things that you can study from a skull. So that's why the skulls are the most important piece that we collect from any carcass that we work on. However, it is good to have whole skeletons because sometimes there are pieces that are not skulls that people need to identify. And so this is an entire sea otter in a box. Every single bone gets the catalog number on it. And that's so that, well, if the boxes, God forbid, fall off in an earthquake, we can put everything back in the right place. I think I'll take a vacation if that happens. But also, archaeologists tend to use the entire skeleton and different skeletal elements in their work. So they were doing a dig in the Transbay Terminal area here in San Francisco. They found some bones. They suspected what they were. They brought them here. They pulled out a scapula from a bunch of different animals that could have been possibilities. And they confirmed that indeed they were sea otter bones. So they found sea otter bones in a midden, which is like a historic trash heap in the Transbay Terminal area. So we want to make sure those bones get back in the right box. And then this sea otter is special because, I have to show this in the right place. Can you see at the tip of my finger, there's a little protrusion, little thing protruding from the skull. That is not natural. That is a piece of shark tooth that is embedded in the skull. So this animal was bitten by a shark, and actually currently about 50% of the southern sea otters that found dead have signs of shark interaction. I'm going to change back to myself and talk to you so that I'm not, I feel like I'm a little shaky this morning. I probably had a little too much coffee. When I first started, it was only about 30% of the sea otters that died were bitten by sharks. Woo. I'm sorry, my light is going nuts. Now it's about 50%. And the reason that is southern sea otters and northern elephant seals both went through a bottleneck in their species. So back in the early 1900s, southern sea otters, people thought they were extinct until they found a small population off the Big Sur coast of 50 animals. Since then, they have reproduced and expanded their range again. And now there are about 3,000 southern sea otters. They're still listed as threatened under the Endangered Species Act. They have to reach a certain number, like 3,100 for 3 years in a row before they can be considered for delisting. So southern sea otters are a protected species still at this time. And because of those shark injuries, they're having a really hard time expanding past their range, because at the northern extent of their range is Año Nuevo Island. And it's there that there's lots of sharks that kind of hang out and wait for marine mammals to come by and chomp on them. So southern sea otters are still trying to recover. Northern elephant seals, however, have recovered very nicely from their bottleneck. They got down to about 100 animals back in the early 1900s, and they're now well up over 225,000 northern elephant seals. And now partly that's because they have a completely different reproductive strategy than southern sea otters. Southern sea otters are pretty much monogamous. They raise typically one pup per year. Mothers put a lot of time and effort into raising that pup, months and months and lots of feeding. Whereas northern elephant seals, they are, now I'm forgetting the word. What is the word, Sarah, when you go, is it colony? Oh, my goodness. They're colonial breeders, or they have harems. A lot of people don't like that word.
Yeah,
But it's the harbor seal colony. Is it called the harbor seal and the elephant seal colony?
Yes.
Okay. I don't know why my brain couldn't put that together, but you have one male and many females, and then the females give birth to the young. They only spend a couple of months, up to 4 months, feeding the young elephant seals, and then they leave and they wean the elephant seals. So we have a lot more elephant seals reproducing successfully than we do southern sea otters. Now, a great white shark is also much more interested in eating an elephant seal, especially a big fat wiener pup, than it is in eating a southern sea otter. Southern sea otters have 1 million hairs per square inch, and they don't have a blubber layer, so they're not very energetic, or they don't provide enough energy for sharks, whereas elephant seals do. So as the elephant seal population has increased, the great white shark population has increased, and thus there's more pressure on the southern sea otters, because sharks just bite them and let go and don't actually feed on them. There's no scientific evidence of partial parts or whole parts of southern sea otters actually being ingested in great white sharks. They bite, they let go, similar to with humans. If they bite a human by accident, they usually don't consume the entire human. I think we probably would have heard about that, but they will eat the northern elephant seals. I'm going to move again. You guys can ask more questions. I also have to get a new glove because my glove broke.
We noticed that. Hey, we do have a question about what you were just talking about, which is what was the former northernmost range of the southern sea otter, and should they be transported north of the shark zone to expedite range expansion?
Oh, and I'm sure you guys have talked a lot about that up in Mendocino, because I know you would love to get southern sea otters up there to help the kelp forest stay healthy. In fact, if you want to look back, there is a southern sea otter night school recording on the Academy's night school webpage that we did back in September, where I showed some other specimens of southern sea otters. And a couple of researchers who actually work on reintroduction also talked that evening. But it's really difficult. Well, their previous range probably was all the way up to Alaska. We think that southern sea otters and northern sea otters shared a range at some point in their history. And then they got wiped out in the hunting trade, which is why we ended up with this big gap in between the two species now. There was a northern sea otter in Mendocino not that long ago. You've had some southern, or maybe it was in Humboldt. You've had some southern sea otters up there, but only like individual young males that are kind of going off and searching. They're really faithful to their natal areas, unfortunately. And so that causes problems for them, because they're not that, would not be that easy for them to expand northward. And then now that San Francisco Bay is cleaner and we have harbor porpoises and bottlenose dolphins coming into the bay, there's hopes that southern sea otters could live in the bay and could succeed, but there's no plans right now for reintroduction, because it's a really tricky process, and you don't want to move an animal and then have it get killed for other reasons. But we are very lucky in this area, because we get to see so many different species of pinnipeds, and we have sea otters, and we have a lot of cetaceans too. So I'm in the cetacean area. I already showed you whales. I'm going to show you a porpoise downstairs, and I want to make sure we get downstairs because downstairs is very cool. And I've talked a lot more than I anticipated I would. I should have known better. But I want to show you this specimen because this specimen is pretty amazing. And I love showing this one. Because where else in the world will any of us ever get to see a Ganges river dolphin? So this, let me put it up on the box. That's a better view, huh? Isn't that incredible looking? Just absolutely stunning. This one came from Pakistan in 1968. And Ganges river dolphins, so most cetaceans have a melon, most tooth cetaceans, odontocetes. So not the baleen whales do not echolocate. We're not even 100% sure how they hear, but animals that do echolocate are the toothed whales, so odontocetes and their relatives like the Ganges river dolphins. They have a melon on the top of their head. They send a signal out of the melon, an echo. The signal comes back, well, it hits the fish, bounces off, comes back to the lower jaw, travels along the lower jaw, which has a gelatinous material in it for conducting sound, to the back of the mandibles, right at the base of the ear bone. So this one has one ear bone on the side of the skull that's closest to the box. I'm a little nervous about letting go, but I'm gonna let go. This is the ear bone right there. So you can see how that lower jaw comes right in, right near the ear bone. So the sound goes right into the ear bone, and then the brain processes the sound and the animal finds its food. Now, Ganges river dolphins have this bony structure that goes around their melon. So their melon's actually inside. And it's kind of like when you cup your hands by your mouth when you yell. If you want to yell really loud across the room, you might cup both your hands around your mouth to direct your sound further. That's what this bony apparatus does. And it has a name, and I've forgotten it again. And then the reason why they're so dependent on their echolocation is this is where their eyes are. They have teeny tiny eyes. And that makes sense because these guys are an ambush predator, just like crocodiles and gars, the fish. They live in very murky waters, so they're not going to be using their vision very much. They're going to sit and wait, and then when they echolocate and find the fish, then they're going to just grab them with those teeth. And if you put this side by side with a crocodile skull and an alligator gar skull, it's amazing to see these three very different groups of animals have evolved similar feeding strategies and similar structures. So it's a great example of convergent evolution. But this specimen is just absolutely amazing, one of my faves. And then there's one other part about Ganges river dolphins that's interesting. So most cetaceans have completely fused cervical vertebrae. So they cannot move their heads at all. Their head is basically fused to their neck. And that helps with streamline so that they can swim faster. They have a nice streamlined body shape. But river dolphins, because of their feeding strategy, actually, and you can see how this one is kind of curved. So these are the cervical vertebrae. There are 7, just like there are in your neck, just like there are in harbor porpoise's neck, and just like there are in a giraffe's neck. 7 cervical vertebrae in mammals. And you can see here that they are individual, 1, 2, 3, 4, 5, 6, 7. And makes it so that they can actually move their neck, which is pretty unique in the cetacean realm. I'll show you a short fin pilot whale cervical vertebrae after I put this one back. And you'll see what I mean by compressed. Any questions? Whoops. Any questions while I roll? Actually, you might want to watch. You can watch while I do this and then ask the question too. So our compactors are like at a big library where you roll each section to make an aisle to get to the other shelf. Go ahead if anyone has any questions.
So Karen asked a general question on how you process skeletons like that sea otter. You or Sarah can talk about that, because I just watched Sarah doing that the other 2 days ago at our facility. But
yeah,
we don't talk
Sarah,
about
you want to talk about that?
Absolutely. So we have some tanks, and we have warm water maceration. So we're going to put like in the sea otter that we're soaking right now, it's been in warm water maceration for a period of time. And now we were joking about how much hair it had, because even though we flensed it down and got rid of most of the skin, there was so much hair still with those bones. And we were kind of joking about that. But we'll take it out of that warm water maceration, and then we will just brighten it a little bit with sodium perborate. And that's all done out near the wastewater treatment plant. And we can maybe have a behind the scenes tour someday and go out there and see what we're doing. There at the Cal Academy, they have a fabulous lab where they can turn the nozzle of a spigot and hot water comes out. But big maceration tanks, right?
Yeah, and we also do a lot of stuff in buckets too, because it's just a little easier. But for things like cetaceans, which are a little bit more difficult to clean because all of their teeth are, I'll show you on the harbor porpoise downstairs, they're called homodonts. All of their teeth are exactly the same, and they just fall out similar to the elephant seal. They're a pain in the butt to prepare because of their teeth. So we will use dermestid beetles to prepare the cetaceans. We'll turn them over and let the beetles do all the work and take it out before the beetles have time to pull all the teeth out. And we use dermestid beetles, which are flesh-eating beetles that clean up the muscle on very small mammal and bird skeletons. So we have the opportunity to use those. So I wanted to show you this short fin pilot whale vertebrae, cervical vertebrae. So this is not one, this is 7 vertebrae fused together. The head is here. And then if you count with me, maybe I should use the camera, but is that all right there? Let's see if I can turn it so that you can see. So there's 1, 2, 3, 4, 5, 6, 7 cervical vertebrae. And you can see how they're all fused together. So typical to most other cetaceans, the toothed whales, they're totally fused together like that. And that keeps them from turning their heads. So some of the other species that can actually move their heads are narwhals and belugas, and then the river dolphins too. So that's about it for this room. I am going to pass the baton over to Sarah, who is going to share some of the Noyo Center's collection while I move down to our other collection room here at the museum that has some more mammals. So,
all right, great, Bo.
Yeah, and you can field some of the questions if I take too long, but hopefully it'll be fast.
We'll
All right,
miss
I'll see you all in a few minutes.
All right, thank you. Wonderful, Mo.
Yep.
So hopefully you'll be seeing my screen here. I'm still seeing Mo. Sheila, you want to let me know if you're seeing me?
I'm seeing you, but did you pin, if anybody pinned Mo, you'll have to unpin and then get back to speaker view to see Sarah.
Great. Good. So this collection is just so incredible. And this relationship between Noyo Center and the California Academy of Sciences has grown through the years. And they didn't used to house very many Mendocino animals. So now it's really exciting to say that there are Mendocino animals. There's a representation of porpoise, dolphin, sea lion, stellar sea lion, quite a few different marine mammals that are now within that collection down at California Academy of Sciences. So in future years, and you want to go see animals from Mendocino, you could see some of our specimens. So we're in the we call it this.
We even have a harbor porpoise. Our articulated harbor porpoise is still on the floor at Cal Academy, right?
Yeah, exactly. If you were to go down and take in their big floor exhibit that is up right now, part of their exhibit of giants of land and sea, incredible exhibit at California Academy of Sciences. It's been up now for 2 years and it's still up, and our beautifully articulated little harbor porpoise is down there to represent Mendocino County animals. So we do get a lot of these same specimens, not any sea otters. It could happen, right? But we collect the specimens, and most of them do go straight down to California Academy of Sciences. Some we will keep up here. And this is, we call this the sea cave, this back room here where I have my freezer. And most recently, super exciting, I don't want to show you too much, but we have just been joined by 50 or 54 or so new specimens that actually came to us from the California Academy of Sciences. And we will be exhibiting them for, they're on loan, and these are the Ray Bandar specimens. So this is a California sea lion that was collected by Ray Bandar down south.
So Ray Bandar was that amazing volunteer that Mo mentioned before that collected thousands of skeletons that they're still trying to catalog and enter into their collection, and they're loaning us quite a few of them.
Yeah, so stay tuned for that exhibit to come up soon. But I just wanted to thank all of you that have reported animals to us through the hotline, and keep on doing that. It's just so important to have a representation. When I go out on the beach and go to, say, a California sea lion that gets reported, I'll go out and I'll collect vibraceae for the whiskers. This is from a little sea lion that stranded fairly recently at Jug Handle and was reported to me by state parks. And I collected the skull. It's probably going to look somewhat like this. This is a young California sea lion. He doesn't have that sagittal crest just yet. But anyway, this is a Ray Bandar, but the one that I just collected is similar to this one, and we'll go down to the California Academy of Sciences. I will also collect some tissue from every animal and put it in this etho alcohol. So every specimen that I collect, well, I'll grab a little bit of tissue, be it from a whale or a seal or a dolphin or whatever, and that is preserved in alcohol and then given to them. Really beautiful specimens that have just arrived. This is something that I'm so excited to have up here. We have on our main floor, we have a stellar sea lion female, but we've never had a male strand up here. So we've borrowed this fantastically beautiful adult male stellar sea lion. He's just massive. And again, this is a Ray Bandar collection piece, but I don't know if you can take into your brain the difference in size of a male California sea lion compared to a male Stellar sea lion. They're massive. They're like huge. And if you see that one mixed in with the California sea lions on the haul out rocks, you'll just be blown away because it looks like a mountain out there. So we don't have as many Stellar sea lions up here as we do California sea lions. We also got some Easter eggs. I'm not going to tell you what they are yet, but we've got a few skulls that we're going to exhibit that perhaps you'll have to look a little closer. What I've noticed just looking at these skulls as I receive them and check them in, every single sea lion has a different look about his face. So some have longer snouts and some have shorter snouts. It's just really, really, really cool to get to know these specimens. So just as a reminder, please, please, please report those dead marine mammals. And if you come across something that you know, I put a green twine around a flipper, I'll attach green twine to specimens that are left in the field. I'll like collect the skull, the baculum, the whiskers, and the tissue, and I'll leave the rest out there for the turkey vultures and for whatever to return it to the sea. But look for that green twine. Also, don't be afraid to report it twice. It's totally fine if you report it. If I get the reports on the animals a number of times, even if it's just, I'll really do some sleuthing. If it's like a fur seal that's been there for weeks and it's just leatherized tissue, it's still really important. We can identify to species by looking at teeth or fur or, you know, getting the measurements. Any little bit of data is really important to this collection. So it looks like our chat line is lighting up, and I can try to answer some questions that pertain to me, but then when Mo rejoins us, maybe we'll address answers at the very end before we let her go.
Mostly we have put in the chat both the link to Cal Academy's live streams where they have a whole host of amazing things, including the Sea Otter nightlife presentation Mo mentioned. But we also put up in the chat Ray Bandar's an amazing video on YouTube of Ray Bandar himself and his collection that we highly recommend you watching. It's incredibly entertaining. You get to see the man and the legend of Ray Bandar.
Definitely watch that. I went down to pick up some specimens for us here at Noyo and do a transfer. So I'll take specimens down from the freezer and then bring stuff back. And I had the honor of seeing Ray Bandar's basement with Mo and Sue Pemberton. And, you know, these exhibits in this work are absolutely just so amazing to see. My mind has been blown. It's imprinted on my brain how, you know, just the work, the body of work, that collection, going into the back rooms there and seeing these beautifully. I mean, it's an art too, right? That everyone is so beautifully preserved. They're not bleached, they're not painted, they're not stuck with glue. They're treated really nicely, and their preservation forever is really important.
Can you show the notes he writes on the bottom of them that where they were collected and what? Oh, that one doesn't seem to have it. Oh, yeah, it does. You just can't read it.
Oh, there it is. Yeah. I don't know if you can see. I'll try to get that. But right in here, his writing is like immaculately perfect and tiny, and it says everything. So it says where this animal stranded. It says that it's a California sea lion. Each individual specimen has a unique identifying number for the collection, and it says where this was from Goat Rock Beach, so south. It died July 23rd, 2001. And its standard length is a measurement of the length of the animal. So this animal here was 224 centimeters. So not quite an adult California sea lion, but getting that standard length is the tip of the nose to the tip of the tail, not to the tip of the flippers, but that standard length on a dolphin would be tip of the snout to the notch in the tail. And I'm training and working with our beach response team up here. So we do have a team consisting of Wendy, Leah, Karen, and myself. and Sue Coulter and different people up here who have shown interest in helping respond to these animals. And we don't always collect specimens. Sometimes we will just collect the data, tissue, measurements, things like that.
Well, look, it's great to see your sea cave, Sarah, and thanks so much for giving us a preview, but everybody will hopefully get to see this skull wall on exhibit as soon as we open the downtown center. We're hoping in early May, so keep your anticipation building for that. It looks like we got Mo back, so I think we can swing it back over to Mo if you want to pin her video again.
I have to do the same. Can you guys hear me all right?
Yep.
Okay. That worked.
Thanks, Sarah.
Thank you, Sarah. Yeah, that skull wall, we have over 400 of raised sea lions up on our skull wall here in the museum. And when the first skulls exhibit went up in 2003, there were over 800 sea lions on the skull wall. So my first day of work in 2002, February 22nd, 2002.
You washed up on the
That's
beach.
the one you took off the beach.
Yes. 6 years
What
ago.
was that?
I think that was somebody who hadn't muted themselves. Can you please?
Oh, I was like, no, my first day of work was not on the beach. I don't know how you know, but my first day of work was in Ray Bandar's collection, starting to package up California sea lion skulls for the skull wall for the skulls exhibit. So that was quite an amazing first day, and I'm so pleased, and Ray would be too, to know that the sea lion wall, an iteration of the sea lion wall, is going to now exist up in Fort Bragg as well. So I hope you all enjoy it. All right, we are down in our oversized collection room, also called the Antler Room, which the entomology team refers to as the Weevil Room, because they have part of this room where they store their weevils. When we moved into the new museum in 2008, we realized we did not have enough space upstairs for all of our specimens. The entomology department said, oh, we have too much space in this weird room downstairs for our weevils, so you can have some of it. And we took 3/4 of the room, and they have a small section still. But you know, weevils are teeny, teeny, tiny, and our specimens are much bigger. So this room sort of makes sense because we have some ungulates, like that big eland up on the wall mount, and some other horned and antlered specimens around the corner. So the fact that ungulates and cetaceans are somewhat related doesn't make it completely obscene that they're in the same room together, but mostly they're in this room together because we needed space. But taxonomically, it kind of makes sense. So we are, except for this one that I'm going to show you first, because this one actually belongs way at the beginning of the taxonomy, next to elephants and kangaroos. This is, I'm going to take the sign down, and there'll be a big reveal of the sign later. This is a Dugong skeleton, and this is a complete skeleton. There's the head, there's the rib cage, moving all the way down. There's some pelvic bones. To the tail. And then back again. So dugongs and manatees are more closely related to elephants and kangaroos than they are to any other marine mammal species. And that one key characteristic that those groups all share, and it's not very easy to see here, but they have molars. So dugongs have some teeth in the front here, but they have molars in the back, in their lower jaw, and I'm not sure if they have them in their upper jaw. Manatees have them in their upper jaw, as well. But these molars start growing up inside the bone back here, and then they move forward. They're called marching molars or a conveyor belt. And what happens with these herbivores, I can just barely see a molar in there. But what happens is the molar in front gets worn down from eating a lot of vegetation and the sand and stuff that gets in the vegetation. When it's worn down, it falls out, and the molars behind move in to take its place. So that's the conveyor belt like teeth that are part of these species. And so that allows manatees and dugongs to replace those worn down teeth. Other species in the world, in the whole animal kingdom that have those polyphyodont teeth are reptiles and crocodiles and things like that, and some other mammals have they have teeth that continuously grow, one set that continuously grows, but is similar but not exactly the same thing. Rodents do that. But as in humans, we only have two sets of teeth. We have our baby teeth and we have our adult teeth, and that's it. But these guys will continuously grow teeth throughout their lifetime. Now, elephants have a finite number of molars that can be replaced. And once they get to the end of the tooth buds, when the tooth buds have all been used up, then they can't survive anymore. They end up dying because they can't feed themselves. But that's about 83 years or something. So this dugong is from Sydney, Australia. And I don't know if you can read that date here, but it's December 11th, 1893. So this is one of the older specimens in our collection. It was not collected by anyone at the Academy. And in fact, a lot of our specimens are from after 1906 that we collected. However, prior to 1906, we received specimens from orphan collections. So in the 1906 earthquake in San Francisco, the fires destroyed everything that we had in the Academy collection. We estimate there probably were about 25,000 specimens in the collection, but there's no way to know because the catalogs also burned in the fire. And so we had to rebuild the collection starting in 1906, but we received collections from other people. And in this case, this dugong came from the Stanford collection to join our collection. So we do have some older specimens. They just were not necessarily part of the original Academy collection. So are there any questions while I move?
Let's see, Sheila had to jump off, so I will try to field these questions. People are commenting on the crazy shape of that dugong without a mouth.
I know, they're really kind of amazing.
That's
like shape, it
mind-boggling.
goes downward for feeding. It's really quite stunning.
Here's
Hold
a
on, I'm
question
having
though.
some
Has
cable issues.
genetic testing confirmed the morphologically based phylogenetic relationship? So genetic testing
Yes,
to
for the
Yes.
dugongs, manatees, and elephants.
Mhm.
Yeah, people have done phylogenetic trees using genetic testing. So it's not just the morphological similarity.
Nice.
They are actually relative. Whoopsie.
Thanks for that question, Lawrence. The chat, I don't have my glasses on. Multitasking, looking at tiny screens and big screens, multitasking and all of that. But if you have more questions, now's the time. I know we've gone over an hour, but I have a feeling that most of our audience would probably sit here and talk to you all day, Mo.
Well, I'll go a little bit longer. There's not a ton of stuff to show in this room. There are a few key items that I want to share, but certainly feel free if you need to get lunch or something to step
Yes.
off. And it is recorded, and I know Sheila said it would be shared, so you can always watch the end later. I just got a reminder that my dog needs to be walked. We'll do that in a little bit. All right, let me share a few more specimens here with you. You guys all know the beautiful orca that is up your way. And do you know the ecotype of that orca? Somebody knows. Somebody must know. Anybody want to put it in the chat?
Feel free to put that in the chat if you'd like to, folks. The ecotype.
Let me start talking about ecotypes while you do that. So there are 3 ecotypes of orcas that are found along the California coast. There is the residents. We'll look over here. So here, this is an orca jaw, lower jaw upside down. This is the cranium this is the roof of the mouth. So we're looking at it upside down as well. The only way we store them. This is a resident orca. So this is an orca that was captured in 1968 from Washington State, and actually was a member of the Naval Undersea Research Orca team in Hawaii from 1968 to 1971. I guess at which point it died, and we received the specimen here at the Academy. So because this is a resident from Washington, it eats fish, primarily salmon, and thus you can see the nice pointy teeth. I saw in the chat someone said transient, and that's right. Your orca is transient from the Alaska region that feeds on marine mammals. So your orca also has these very sharp, pointy teeth. And they're not worn down at all. They feed on, some transients feed on gray whales and other larger whale calves. And then your orca in particular had, I think it was 5 harbor seals in its stomach, or sea lions. Do you remember, Sarah?
Yeah, so our orca, it's funny whenever we say it's 5 or 6 harbor seals in its belly, which we were discussing the teeth when someone pulled out stomach contents, and they were harbor seals. And so he'd been feeding really recently right off of our coast, and it was harbor seal pup time. He had a number of pups. So that kind of answered the question about the teeth, because they were slightly worn.
Yes, so this one is also slightly worn in the front. I remember before we got to the stomach contents, I actually was examining the blowhole and the throat to see if it had drowned, because sometimes you get blood in the blowhole. And I found hair, and it totally freaked me out, because I was like, oh my God, this cetacean has hair in its throat. And then once, my brain did not automatically go to food. It was like, why is there hair in this throat? But it was because of the harbor seals that were in there. So this specimen, along with an orca that we have here on display that is articulated as a complete skeleton, are both a different ecotype. They are the offshore ecotype. And as you can see here, this female's teeth are worn completely down, even to the point where there's abscesses on both sides of this upper jaw. And the same thing is the case with our orca that's upstairs. He was identified during life as an offshore ecotype. They had known him for several years, and he was aged at 12 years old. So when we first walked up to him on the beach and saw these teeth, his teeth were worn down to the gum line, we immediately thought it was an old man, but his dorsal fin was not tall enough. His body was not big enough. He was only a juvenile, and yet his teeth were already so worn down. And the reason why offshore orcas have worn down teeth like this is because they primarily feed on sharks. And sharks have dermal denticles on their skin, which is kind of like sandpaper. And so it wears down their teeth. Now, it brings up other questions when you run across an animal on the beach who's a juvenile, he's not sexually mature, his teeth are completely worn down to the gums to the point of abscesses, which had to be painful. Like, how is that a good feeding strategy for a species or for an ecotype? And we don't really know. And that's one of the very cool things about science that just keeps me coming to work every day, is you end up with all these questions. You find something new and then you get some more questions. Let me take a quick glance. There's my computer down here. I just want to show you the lower jaws from a Baird's beaked whale. So Baird's beaked whale, this is an adult male. They only have 4 teeth in their lower jaws, 2 of them that protrude above the gums. And in the male, they're quite large. In the female, they're very small and hidden under the gums. So keep that in your mind for a second. And then this model down here, oops, I'll have to take it out. Let me pull it out and bring it over. Let me move myself one more time. Or well, twice. We'll actually do 2 moves, but 1 move here. And I'm going to show, you can see our harbor porpoise in the background.
Is that yours or is that ours? Ours
That's
is
ours. This is
yeah.
ours. This is the harbor porpoise puzzle
Oh, right. Very
that
cool.
goes out with the since we're trying to keep public programming from getting big crowds, it's not going out yet on the main floor. So this is a model of what a Baird's beaked whale looks like. So there's, you can see the nice melon where it produces its sound, shoots the sound out, the sound comes back in the lower jaw and goes back to the skull. But this is a model of a Baird's beaked whale. So back in 1925 in Santa Cruz, something washed up on the beach and everyone was quite puzzled. Oh no, now I just lost my, hold on a second. I have to get back to you guys. Share. So on the beach, this crazy looking creature washed up. And if you could see this whole picture, this gentleman, can you see my cursor? This gentleman right here is carrying a shotgun. Yeah, so he was obviously very nervous about this creature. But it has a very big bulbous head and what looks like a neck, and the whole body is like a long, skinny body. So people thought it was a sea monster. So it was called the Moore's Beach monster or the Santa Cruz sea monster. But then another guy named E.B. Wallace came down and he said, no, that is not a sea monster. That's actually a plesiosaur that was probably frozen in a glacier, and the glacier melted and then it came down onto the beach. And then scientists at Cal Academy got wind of this from the newspapers and they said, oh, we better go down and look. And they went down and a specialist from the Academy put everything to rest and said, no, this is a Baird's beaked whale. And, you know, they're a very pelagic species. They're very rare. You don't see them very often. But it is not a plesiosaur and it's not a sea monster. It is actually a beaked whale. There are still people out there that believe that it was a sea monster. Stop sharing. However, and they think that that I am actually hiding the Santa Cruz sea monster here at the Academy. So this is that exact specimen. Again, it's upside down, sitting on the top of the head. So you're looking at the roof of the mouth, and then there's one of its mandibles next door to it. So this is the first Baird's beaked whale that was added to our collection in 1925. Since then, we have added 3 of them to the collection, including one in 2003, which was the jaw that I just showed you, and one in 2016 that is still buried outside, getting cleaned. And so I need to unbury that sometime soon. I just got a little worried about it the other day because I don't know how long bones stay together when they're still in the ground, but it's about time to get it out of the ground. All right, sorry about that. I thought I could move with the camera on and not have to turn it off, but I realized that's not a good idea. So let's just move a little bit closer to the harbor porpoise. Good Lord, no. So I can give you a close-up look. So this is our harbor porpoise puzzle. So it actually comes apart in pieces so that our public programs team can share it with our guests. Aha, the zygomatic arch. Maybe it didn't exist there. So here I really wanted to show you again those fused cervical vertebrae. So here are the 7 cervical vertebrae of the harbor porpoise. They are not attached to the skull. They are actually, and these are supposed to be glued in there. We have to do some maintenance on this before it goes back on the main floor. But you can see that they are all 7 of them fused together. And then here are these small teeth that I talked about. Let's see if we can get a close-up of the teeth. Can you see how all of those teeth look exactly the same? They're like little spatulas. So it is very difficult to put those teeth back in the specimen. And harbor porpoises and most cetaceans do not, they don't actually chew their food. They just kind of suck them in. And so that's why they do use their teeth to grasp them, however. And then the other part of this harbor porpoise that I really love is the tail, which also comes off of the puzzle. But doesn't it look like rattlesnake tails, those little teeny tiny little bones that go all the way down? So the flukes, as you can see from the skeleton and any cetacean skeleton, they have a tail of vertebrae. They do not have bones in the flukes. The flukes do not have any bones. A lot of people get confused by that. And they think that our cetacean skeletons are dinosaurs because of their tails. So let me put this head back in. Any questions while I do that?
I didn't have any questions, but I did have some fun comments about our Laura, who's in the front of this house, sitting right near the orca, looking at his teeth. And
oh, nice.
And
they are indeed pretty pointy, aren't they?
Pretty pointy. And that feeding strategy, so the orca and that harbor porpoise, that sucking in and swallowing and the feeding strategies of these marine mammals is just fascinating. And you spoke about the Baird's beaked whale, and I'm just trying to wrap my brain about it's down there in the dark and it's feeding on mostly squid, and those teeth would be used to just sort of pin it in place, so to speak.
No.
No.
And on those And on those, is that my sound doing that?
I don't hear anything.
Oh, I hear a very weird beeping sound in my head.
Uh-oh.
So we'll have to make this quick before something explodes in my ear. No, they are for sexual selection, actually. In that species, they do not actually use them for grasping the fish.
Okay.
It's mostly for showing off who's the best male of all, right?
Okay. Very cool. I'm glad I asked because I was way off.
Nobody else hears that weird noise, huh?
I don't hear it. Back to the monster, it's just whenever there's a blob of something that washes up, the detective work around all of these specimens is just endlessly fascinating. And when I'm out and find a blob somewhere, I can call Sue Pemberton or Mo and chat with them about trying to figure it out. And I think it's probably one of the most favorite things that I am doing in my life right now.
I figured out what that was. The camera.
The camera's beeping, so it's about. Well,
all right. One last section of this room. So on this, well, behind me, those are all the Weevil cases. So you can see they have all their teeny tiny little weevils in there. But on this rack here, at the end of the horned and antlered specimens, we actually have a bunch of whale bones, blue whale ribs, a humpback whale lower jaw. On the bottom there, that really gross, dirty one. And then I have a very special specimen to pull out for you. Let me grab it. So this gives you an idea of the size. I'm 5'7", 5'8". So it's, 5 foot, over 5 feet long. This is a narwhal tusk. And you can you see kind of the curve, the beautiful curved pattern of the tusk? So this is one of the narwhal's canine teeth. So narwhals do have typically 2 tooth buds that are canine teeth to make sure I don't hit anything in the ceiling. That's the end of it. And those canine teeth, only one of them will actually grow into a tusk like this. And that tusk is usually the left one. For some reason, the left canine tooth grows long. There have been some cases of a narwhal growing 2 of them long or one a little bit longer than the other. But for the most part, it's the left tusk that grows very long. Now, narwhals are also not using this tusk for catching fish, because if you caught a foot, like people originally thought they speared their fish with them. If you speared a fish here, your mouth is 5 feet back. It's not really going to work. They could be sexual selection, again, like the long tusks showing the male or the female who the fittest males are. Same with elephants. Elephants get the really large tusks as sexual selection. It could be in dominance, but there has not been any photo evidence or anything of narwhals jousting with each other with their tusks to, show dominance. But what some researchers did discover in the last few years with some drone footage is narwhals swimming in a pot of fish and swinging their tusk and stunning the fish and then sucking the fish up to eat them. So that is the current belief or theory about the purpose of the narwhal tusk. This one, we don't know exactly where it came from. It was donated to the collection in 1978. And along with it, there was a letter from an art curator at the Smithsonian from 1951 to an individual who had a narwhal tusk in their private collection. And the Smithsonian was basically like trying to get them to donate their private collection narwhal tusk to the Smithsonian. I assume because it's in the file with this, that letter actually came with this. So this could be that specimen that was held in a private collection and then donated to the Academy in 1978. So we only have 2 real narwhal tusks. We have one cast one. Actually, we have 2 cast ones now because we have a cast skull with the tusk as well in it. So that's a pretty special treat to end our walk through the marine mammal collection here at the Academy.
Wow. Just wow. What an incredible place you work. I've had the pleasure of stopping by a number of times with my daughter who's studying marine science, and Mo showed her the Chihuahua skull that is very different from the St. Bernard skull, not just in size, but dogs have every different shape of skull. But really fascinating just to learn about the different adaptations and, you know, why does the sea lion have that sagittal crest, and, you know, why doesn't the stellar sea lion have all these questions? That's, like you said earlier, the beauty of science is that it brings forth so many questions. And
absolutely,
how special these specimens are, that we can see them and share them and learn from you and all of that. So any other questions? I see, let me just check the chat real quick. Thank you, fabulous. Everybody just loves and adores you, Mo. Thank you so much for taking time in your day.
You can probably tell that one of my favorite things to do is show off the collection. Why? What we think is going to be an hour tour ends up being much, much longer than that.
Yeah. Well, hopefully next time we'll get you up here in person and hang out with you in person, and cross our fingers that not a lot of whales wash in over the next few months, and that the UME kind of tapers down a little bit. Get onto these websites and look at some of these talks, people. It's just a treasure trove of scientists sharing their information for us. The upside of COVID is this experience. I don't have any more questions for you, Mo, but I do have just lots of appreciation. And for you Noyo Center folks, before we let Mo go and walk her dog and enjoy the rest of her day outside a little bit, there will be one more science talk that's part of our Whale Festival celebration virtual. Becky Jewell, who rode her bicycle from Alaska down to Baja kind of followed the gray whales a few years back. So she'll join us on the 31st at noon, another midday talk, but she's terrific. She was looking for gray whales, but she was also looking for cake. So she's a really fun person. We're super excited to have her join us, and hope to see many of you back there with us again. And donate if you can. Become members of Noyo Center or California Academy of Sciences, and keep reporting those dead marine mammals and carry on.
Sarah, did you say she was in search of cake? C-A-K-E?
Yes.
Oh,
yeah.
I think I might have to join on.
I know. She was, yes, in pursuit of gray whales and also really good cake. So she's super fun, and she stopped by on her very heavy bicycle. She had encountered bears in Alaska and people along the coast, and, you know, stopped in Fort Bragg and came in with her bicycle and looked at the orca and carried on and broke spokes on the hills south of here and checked in with the Mercers. And yeah, anyway, she'll have some great stories for us.
She was very excited that we got hold of her. She remembered the Noyo Center and really enjoyed her visit here. She
Oh,
actually
great.
is in Manchester, England, so it's noon here, but it's gonna be like 8 o'clock at night for her. So,
Oh, great.
so it's
Well,
tricky
that's-
finding a time, you know.
But that's the perfect time for a lunchtime seminar. So there
Yeah.
we go. I'll
We'll
have
have
to
to
put it
bring
on my calendar.
Yeah, and we'll all eat cake together and talk to Becky Jewel, make her mouth water.
Perfect.
right,
Well,
thank-
if you guys come up with any other questions, just send them to Sarah and she'll send them to me. And hopefully we'll see you all in person sometime soon.
All right.
Wonderful.
Thank you.
Thank you.
That was great. Thank you very much.
Thanks.
Thank
Well,
you.
see you
Bye-bye,
soon.
everyone.
Bye.